Good morning, everyone. Can you hear me okay?
We
Can
can hear
you
you.
raise a hand if you can hear me? All right, thank you. All right, good morning. We will continue with our discussion of cell respiration today. I'm going to try to show us a video a little bit later today. We've got some nice ones, glycolysis and citric acid cycle. I'll remind you about the citric acid cycle a little bit later when we get there. And I'm going to try to show us a very short electron transport chain video from the SmartWork animations as well. Reminder for everyone, we have our second exam on Monday at 9:30. We will have 50 minutes for that exam, covering material from chapter 9, looking at evolution of genomes. 11 and 12, we are looking at the structure and function of membranes. In chapter 3, we're looking at energy resources, energy processing in the cell. And then is this chapter, we're looking at where some of the energy comes from in cell respiration, the extraction of the high energy electrons and chemical bonds from our food molecules. So I want to remind everyone to make sure that you have logged into your Reef account and that you have joined our course for our clicker questions today. And we will pick up where we left off on Monday in the mitochondria. We described the process of glycolysis, the production of pyruvate as a product of the 10 steps of glycolysis in the cytoplasm. And we described, or what we'll look at today is what happens to that pyruvate once it has been transported across both of these membranes. We're not going to look at the transport itself, but pyruvate has to be transported across the outer mitochondrial membrane as well as the inner mitochondrial membrane into the matrix where the enzymes reside that will convert pyruvate into acetyl-CoA. That's our first step. And acetyl-CoA will then enter the citric acid cycle to continue the oxidation of that fuel molecule and completely oxidize the carbons that are present in the pyruvate. So that's what we're following is the processing, the oxidation of pyruvate. And I think I mentioned on Monday that other fuel products can also enter the citric acid cycle. Fatty acids, the breakdown products of fatty acids from fat, our other primary fuel source, can also be fed into the citric acid cycle, and electrons can be extracted from those carbons as well. All right, so today, our first step is once that pyruvate has been transported, can everybody make sure that their mics are off? I see one person has a mic on. I'm going to mute you. There you go. We're going to look at the biochemical steps of this phase of cell respiration. So pyruvate, the 3 carbons of the pyruvate, and remember we have 2 pyruvates for every glucose molecule. Pyruvate's going to be oxidized. Pyruvate plus coenzyme A, which we're abbreviating here, is there's coenzyme A with its sulfhydryl group. High energy molecule, or will form actually a high energy molecule, will be converted to acetyl-CoA and release a fully oxidized carbon as carbon dioxide. So in this step, the 3-carbon pyruvate will combine with coenzyme A, and the result will be the formation of acetyl-CoA, a 2-carbon molecule. So what we have is a multi-enzyme complex that carries out 3 reactions. One reaction is the oxidation of pyruvate, removing this carbon and those oxygens, removing that carbon as a fully oxidized carbon released as carbon dioxide. That hydrolysis reaction releases energy that drives the reduction of NAD+ to NADH. Right, so this oxidation drives this reduction, extracting electrons from the pyruvate molecule. They're stored in NADH. We'll see later what happens to those electrons. And that also then drives the condensation of these remaining 2 carbons with coenzyme A forming acetyl-CoA, a high energy intermediate. This thioester bond between this sulfur and this carbon, that thioester bond is a high energy bond, meaning it's very unstable, chemically unstable, and hydrolysis of that bond will drive the next set of reactions. Right, so complicated biochemistry class maybe talks about all of the different steps of this, but for our purposes, an enzyme complex carries out these 3 processes: removing carbon as a carbon dioxide, which is released, diffuses out of the mitochondria, diffuses out of the cells, we breathe it out, right, as a byproduct. Formation of NADH and condensation of the remaining 2 carbons with coenzyme A. And we call that molecule acetyl-CoA because that's an acetyl group there. So what happens to acetyl-CoA? That is a starting reactant that's fed into our citric acid cycle. Citric acid cycle, sometimes called the TCA cycle for tricarboxylic acid, because citrate, which is formed in the first step, is a tricarboxylic acid. There's 1 carboxyl group, there's a second, and there's a third. And what we see here is the 2 carbons that are highlighted in red, the 2 carbons from acetyl-CoA, will combine with 4-carbon oxaloacetate molecule to produce or to form citrate. Citric acid, citrate, the ionized form is called citrate, a tricarboxylic acid. The rest of the cycle will, the next 7 steps, will decompose citrate back to the 4-carbon oxaloacetate, providing more oxaloacetate to be able to combine with more acetyl-CoA, right? Keeping this cycle moving in the forward direction. What does the cycle do? The cycle will completely oxidize the acetyl-CoA, the remaining carbons that originally were in our glucose molecule. Along the way, we will see 1 carbon removed, another carbon removed in these 2 oxidation steps that also produce NADH. We will see another oxidation that extracts electrons and carries away the electrons in another electron carrier called FADH2. And then we have another oxidation at the very end that will regenerate our oxaloacetate. And in 1 step, a little bit of ATP is made. In this illustration, GTP is synthesized, but the hydrolysis of that GTP will actually produce a molecule of ATP. So that's what we're going to look at, all of the steps of the citric acid cycle. And again, you're not required to know the structure of all of the intermediates. I'm not even going to name the enzymes, but I want you to know what are the steps that are driving this, what are all the different steps in this biochemical pathway. So let's start with step 1, the combination of acetyl-CoA. And again, let's follow the carbons to keep track of what's happening, and then look at each step. What is the biochemical process that's occurring? So acetyl-CoA will combine with our 4-carbon oxaloacetate. This condensation step produces citric acid, a 6-carbon molecule. That condensation was driven by hydrolysis that removed coenzyme A, right? Breaking that high energy thioester bond drives that condensation reaction. In the second step, citric acid is isomerized into isocitrate, still 6-carbon molecule. And isocitrate is the substrate that will undergo our first oxidation step. Isocitrate is converted to alpha-ketoglutarate. In that process, 1 carbon has been oxidized. So the fully oxidized carbon is released as carbon dioxide, and the oxidation step produced NADH. The electrons extracted in the oxidation, removed from carbon, are now have been handed over to NADH. So this is the first of 4 oxidation steps in this process. Notice our isocitrate has lost, because it's lost a carbon, has been converted to alpha-ketoglutarate, which is a 5-carbon molecule. In the next step, that alpha-ketoglutarate will be oxidized. Our second oxidation step, electrons transferred to another molecule of NADH. In that process, the 5-carbon alpha-ketoglutarate loses the second and really our last carbon from our original 6-carbon glucose has been released as carbon dioxide. And that is driving the addition of another coenzyme A to form a high energy bond in succinyl-CoA. So alpha-ketoglutarate converted to succinyl-CoA, 4-carbon molecule, because alpha-ketoglutarate, one of its carbons has been removed. And remember also, we're looking at one cycle of the citric acid cycle, right? We're looking at one cycle initiated by pyruvate being converted to acetyl-CoA entering the cycle. So, and we want to also remember that this cycle is running 2 times for every glucose molecule, because each glucose by glycolysis has produced 2 pyruvates. So we've gotten to this step. We have a 4-carbon succinyl-CoA, which will be converted to succinate by a hydrolysis reaction. The hydrolysis reaction removes or cleaves the coenzyme A. That drives the synthesis of GTP. And in most cells, the GTP will be hydrolyzed, which will drive the synthesis of ATP. So this is our ATP synthesis step in citric acid cycle. And if we can look at the combination of what's going on in step 5 on the next slide, the hydrolysis reaction that removes the coenzyme A from succinyl-CoA to produce succinate will drive the phosphorylation of GDP to generate GTP, and then the hydrolysis of GTP will drive the phosphorylation of ADP, and we end up in some cell types, not all, but most cell types, producing ATP. So here we are with our succinate. We have gone through 2 oxidation steps already. We've lost 2 carbons as carbon dioxide. In our step 6, succinate will undergo a 3rd oxidation, succinate converted to fumarate. Electrons extracted from succinate are going to be passed to another energy carrier, FAD, and FAD is reduced to FADH2. FAD is flavin adenine dinucleotide. It's very similar in function as NAD+, which is a nicotinamide adenine dinucleotide. So another electron carrier, carrying a pair of electrons away from that have been extracted from our fuel molecule. Succinate converted to fumarate. Finally, on our last page, that fumarate will then be hydrogenated, meaning hydrogens will be added, converting fumarate to malate, and then malate will undergo our fourth oxidation step as it's converted to oxaloacetate, regenerating oxaloacetate, which can then combine with another molecule of acetyl-CoA. That last oxidation produced another reduced electron carrier, NADH. So remember, every time an oxidation occurs, electrons are extracted from our fuel molecule, electrons are lost from the carbons, transferred to our electron carrier in the form of hydrogen atoms. So we can then revisit our cycle and look at really what we want to remember from the cycle. I want you to know the steps, but remember, what is the purpose of this cycle? Right? What is the cycle producing overall? You could go to, and you can actually follow the movement of each individual atom in the citric acid cycle. If you go to our SmartWork animation, it takes a few minutes, about 8 minutes, and it will show you, for anyone who's interested, all of the individual steps, where the electrons are going, where the atoms are coming from, and where they're going. But for our purposes, what you need to know from my class is what are these steps, all of these 8 steps? What is the output? What's the yield of the citric acid cycle? Right? And so if we think for every pyruvate that has been converted to acetyl-CoA, fed into the citric acid cycle, generating citrate, and then the following 7 steps decomposing that citrate to regenerate oxaloacetate, keep the cycle going. 1 turn of that citric acid cycle will produce 4 high energy reduced electron carriers, right? At this step and another step and our last step, 3 molecules of NADH are produced. At step 6, succinate converted, oxidized to fumarate, and FADH is generated. The first 2 oxidation steps released fully oxidized carbons as carbon dioxide. So overall, 2 carbons are produced as a byproduct. And at one of our steps, succinyl-CoA converted to succinate produces a molecule of ATP. So for each turn of the citric acid cycle, 2 carbon dioxides, 3 NADHs, 1 FADH2, and 1 ATP. But remember that for each molecule of glucose, and that's how we're following the yield, right? The yield of cell respiration. We're always following per molecule or per mole of glucose, our starting material. What does the cell produce? And remember, for each molecule of glucose, the glycolysis is producing 2 molecules of pyruvate. So for each molecule of glucose, we have 2 rounds of the citric acid cycle. So overall, what's being produced is 4 carbon dioxides, 6 NADHs, 2 FADH2s, and 2 ATPs. So what are all of those electrons in those electron carriers? Where are they going? What is the purpose of the citric acid cycle? Right? Why is the cell going through all of this trouble in the matrix of the mitochondria to generate all of those high energy electron carriers? What we're going to see next is all of those electrons carried in the electron carriers will be used to generate ATP in the presence of oxygen. So our next step is transfer of all of those electrons to our electron transport chain. The electrons that are carried in NADH, carried in FADH2. But before we go there, let's see if we can summarize what has been the yield of our respiration so far. And we're talking about our aerobic respiration because the citric acid cycle, as we'll see, requires oxygen to accept, as a final acceptor, accept the electrons being transferred from the electron carriers. So for aerobic respiration, what we'd like to do is take a bit of a pause and summarize what has respiration yielded so far. I'm going to ask you, what has up to this point, completing citric acid cycle, starting with a molecule of glucose, how much ATP has been produced? If anyone wants to tell me with a raised hand or tell me in the chat. All right, 4 ATPs. Where have the ATPs come from so far? From one molecule of glucose, where have the ATPs come from? Correct. We yielded a net of 2 ATPs from glycolysis, right? 2 ATPs are used at the beginning of glycolysis, 4 are generated, a net yield of 2 ATPs. And as we saw in our citric acid cycle, each round of the citric acid cycle yields 1 ATP. We have 2 rounds, 2 turns of the cycle per molecule glucose. The cell has been able to generate 2 more ATP. What else is an important yield? What about the electron carriers? How many NADHs have been produced so far? And where have those NADHs come from? Have we generated any NADH in glycolysis, the beginning of this whole process? Have we generated any from glycolysis? Yes. How many from glycolysis? We've got 2, correct. How many have we, do we have more NADH? Where's the next NADHs come from? What's the next step? After glycolysis has produced pyruvate, what happens to the pyruvate? We're missing a step. Where did we start? Where did we start today? We started with pyruvate today. What happens when pyruvate is oxidized? The first oxidation step of pyruvate, right? That first step generates how many NADHs? Per pyruvate, how about per molecule glucose, right? 2. As the pyruvate's converted, oxidized to acetyl-CoA, 2 more ATPs. Where else do we get NADHs from? After the pyruvate is oxidized to acetyl-CoA, citric acid cycle, how many NADHs? 6. And any other electron carriers generated in the citric acid cycle? Yes. So let's put them all together. From one molecule of glucose, in addition to the ATPs, 2 NADHs from glycolysis, as glycolysis produces pyruvate. For each pyruvate that is converted to acetyl-CoA, 1 NADH. We've got 2 pyruvates, 2 more pairs of electrons are transferred to NADH. And then as we just saw in citric acid cycle, for each round, 3 NADHs, we've got 2 turns and 1 FADH per turn of the citric acid cycle. We've got 2 turns. So we've got a total of 10 NADHs and 2 FADH2s that are being transferred to the electron transport chain. So that was a lot of work that the cell underwent in order to extract electrons, convert and carry away those electrons, carry them away from the fuel and send them to the electron transport chain. So that's what we're going to look at next, is what happens in that electron transport chain. But let's go back for a second here and ask, why is it that this citric acid cycle is driven in the forward direction? Because of course, each one of these is a enzymatically catalyzed reaction, and each of these chemical conversions can occur in either direction. So why is it that this cycle is moving in this forward direction to generate the yield that the cell is going to then utilize in the next stages of cell respiration? Anybody want to think why? In any type of process or biochemical pathway, what would be driving it in the overall spontaneous direction? Overall, one direction, the direction of our arrows, that makes it an overall spontaneous process. Yes, negative free energy. And can we think of how is it? Why is the overall change in free energy negative? Why is overall high energy molecules being converted to low energy molecules? Going downhill, energetically favored, overall process. Remember the concentrations of substrates and products. As long as there's a constant supply of pyruvate, pyruvate will be able to continually combine with oxaloacetate and make citrate. Right? As long as all of these steps are occurring, as long as it's a cycle, as long as oxaloacetate is constantly removed and joined with pyruvate, the cycle goes. As long as there is enough electron carriers, NAD+, right, to be able to extract electrons, as long as there's enough pyruvate. So as long as glucose keeps diffusing into the cell and the glucose undergoes glycolysis, and as long as we have a constant increase in our substrates, right, and a constant removal of the products, then this pathway does not come to equilibrium and it keeps moving forward. Okay. So what happens to all of those electrons that are being extracted? That's what we're going to look at next. But before we do, let's see if we can remember one of the steps in the citric acid cycle. One of the steps we see is an oxidation event that is coupled with reduction of NAD+, meaning electrons are transferred to NAD+ to produce NADH. That reduction is coupled to formation of a high energy thioester bond, and along the way, carbon dioxide is released. So what we're asking here is, where does the sulfhydryl group come from? What provides that sulfhydryl group that is part of that high energy thioester bond? How are we doing? About 5 more seconds? All right, let me close it up. Everybody's got their votes in? What is the one molecule that we've talked about that has a sulfhydryl group, right? That can combine to form a high energy bond. And that would be our coenzyme A. So we've seen coenzyme A being added to and removed from a couple of times, right? Coenzyme A that combined with pyruvate to as part, or the acetyl group of pyruvate, as part of the oxidation of pyruvate, can then be removed. It's added back in later on. So let's go on to what happens next. What happens to all of those electrons that have been extracted in our citric acid cycle? How is the cell generating the ATP that it needs, right? Sure, some cells generate a little bit of ATP out here in the cytoplasm, right? Every cell can undergo glycolysis, generate a little bit of ATP. But all of these biochemical changes that are occurring inside the mitochondria are used to extract, to continue or completely oxidize that pyruvate, extract the energy of those high energy electrons from that food molecule to be able to produce a huge amount of ATP. So what we're going to be looking at are coupled processes. The electrons that have been carried away in NADH, those electrons are going to be passed through an electron transport chain. We're going to see how that is coupled. The movement of these electrons is coupled to the pumping of protons, hydrogen ions, from the one compartment, from the matrix, into the intermembrane space. That pumping of protons creates a gradient. The potential energy of that gradient is a force. Those protons in the compartment where they have been concentrated, those protons can then diffuse through a channel in this complex that we call ATP synthase, and the movement of those protons down their concentration gradient will generate ATP. So that's what we're going to continue looking at, is how transfer of electrons is coupled to pumping of protons that will create a force that will drive the synthesis of ATP. And this is where I recommend anyone who has access to SmartWork that you might want to look at a relatively short 2-minute video of the electron transport chain. And it starts with pyruvate entering the matrix and the electrons being extracted. So I'm going to try to, I'm going to close this down for a second, and I'm going to open SmartWork, and I'd like to show the video. And I realized the last time I showed you a video, there was no audio. I could hear it, but you guys couldn't hear it, because these videos are much more effective when there's audio with them. So the only way I can figure out how to do that is I am going to change my audio to my laptop's audio, and make sure. And I would like you, if you can help me by raising your hand or saying yes once I get this started, and tell me if you can actually hear it. Right? So hold on a second.
The mitochondrion is the site of most of the cell's energy
Can you hear
production.
it? Raise your hand.
After food molecules are processed in the cytosol, they enter the mitochondrion, where they are further broken down. In the citric acid cycle, the molecules are stripped of high-energy electrons, which are donated to carrier molecules, such as NADH. The carrier molecules transfer the high-energy electrons to a chain of proteins called the electron transport chain, which is embedded in the inner mitochondrial membrane. The chain acts as a pump, using the energy of the electrons to move protons from one side of the membrane to the other. The pumping creates a proton gradient across the membrane, which the mitochondrion can tap to make the fuel molecule ATP. The electron transfer begins at a multiprotein complex called the NADH dehydrogenase complex. This complex has a higher affinity for electrons than NADH and easily strips away the high-energy electrons. As the electrons are transferred from one protein to another in the complex, energy is released and used to pump protons across the membrane. Electrons are then transferred to ubiquinone, a different carrier that shuttles them to the next way station called the cytochrome BC1 complex, which again pumps protons as they flow through it. Because each complex in the chain has a higher affinity for the electrons than the previous one, the electrons keep moving through the chain unidirectionally. Finally, cytochrome C delivers the electrons to the cytochrome oxidase complex, a third proton pump. The cycle repeats until the cytochrome oxidase complex has accumulated 4 electrons. From there, they are handed over to molecular oxygen. Oxygen takes up the electrons as it combines with protons, forming water as a product, thereby completing the stepwise path of the combustion of the food molecules.
Okay, so now let me put the audio back to better sound. Hold on a second.
And I imagine everybody can hear me okay again? I imagine that these videos with audio is much more informative. The past 2 times I've showed you a couple of clips, I could hear the audio and I didn't realize that you guys couldn't until I watched the recording. So I hope that encourages more people to actually go to some of these videos that are available in SmartWork, because I think it's really nice summary. So what we're going to do is break down those processes that was just shown to us in this video. So make sure I've got everything back here. There we go. So what we're looking at then is, there we go, okay. What we're looking back then is coupling of electron transport to proton pumping to ATP synthesis. So let's look at the electron transport portion. Our electrons carried in NADH, the reduced form of NADH, are carried in this proton and this hydrogen atom. The transfer of electrons really is the removal of this hydrogen atom plus another electron, which we call our hydride ion. That hydride ion removed from our NADH will then separate into a proton and 2 electrons that are transferred to the next component of the electron transport chain. Right? So our hydride ion removed from NADH results in the reduced form of NADH being converted to the oxidized form of the electron carrier, NAD+. Notice that when the electrons are removed, we end up with a carbon that has a positive charge. That's very unstable and will almost immediately be converted to the final, more stable form of oxidized form of NAD+, in which by the rearrangement of those bonds between the carbons and the nitrogen becomes positively charged. Right? So what we're seeing is the transfer of electrons means those 2 electrons in the bond shared by this hydrogen and this carbon are removed. The electrons are passed to the next component of the electron transport chain, and the proton is released into the aqueous medium. And our NAD electron carrier has been oxidized. It's lost electrons. All right, so that's one step. That is the electron transfer step. So let's ask our second clicker question for the day. Here we're looking at our NADH, a high energy bond, meaning the electrons in that bond have the potential to be transferred to another molecule. So when that bond is cleaved and the electrons are transferred to the next component of electron transport chain, what are the immediate products of that bond cleavage? Right, so what happens when NADH loses loses its hydrogen atom? All right, let me close it up. 5 more seconds. All right, let me close it. The immediate products are NAD plus, oxidized form of NAD plus because it lost electrons, and our hydride ion, right? A proton and 2 electrons. Our hydride ion will be removed. The electrons are passed to a more electronegative acceptor in the chain. The hydrogen ion, the proton, is released into the aqueous solution. So that's what we're going to look at next. This transfer of electrons is coupled with the movement of protons. As we can follow our little pink line, as electrons are moving through these complexes, and we'll talk about those a little bit later. These are protein complexes that have electron acceptors within them. Electrons are transferred from one to the next, eventually combining with oxygen to form H2O. The movement of those electrons results in the movement of protons, hydrogen ions, across the membrane. So notice we're looking at a little schematic of our mitochondria. We have our inner mitochondrial membrane and our outer mitochondrial membrane. This is the matrix. This is the intermembrane space. Protons are moving from the matrix side to the intermembrane space. We end up with a gradient, a higher concentration in the intermembrane space. So electron transfer coupled with pumping of protons. Because this pumping has produced or created a proton gradient, that potential energy in that gradient is a force. The protons can diffuse back across the inner mitochondrial membrane from the intermembrane space to the matrix side. And the only place they can pass through, the only place they can diffuse through is this complex in green here that we call ATP synthase. And the movement of those protons will drive phosphorylation of ADP to generate ATP. And so we can correlate the synthesis of ATP with the loss of electrons from NADH and FADH2. It's the oxidation of those electron carriers that will drive the synthesis of ATP. So if we follow our electrons and look at it in a slightly different way, here is our NADH carrying the electrons that have been extracted from citric acid cycle, as well as even glycolysis from our fuel, right? Our fats, our carbohydrate molecules, high energy electrons, the potential energy of those electrons, the potential of those electrons to move. This is a strong electron donor. NADH, while it will accept electrons from food, it will readily transfer its electrons to the next component of the electron transport chain. And so what we're looking at here are all of those respiratory complexes in the inner membrane. And what we're showing is that each one is more electronegative than the next, meaning that they have a greater affinity for electrons. So this one has greater affinity for electrons than this one, and this one has a greater affinity for electrons than this one, and this one has a greater affinity for electrons than NADH does. So NADH will readily transfer its electrons to the next acceptor in the chain. And those electrons will move down the chain, losing energy along the way. The NADH is oxidized, it loses its electrons. The movement of those electrons will power the pumping, the movement of protons from this side of the membrane, the matrix side, to the other side of the membrane, the intermembrane space. This can only occur, this electron transfer, a little pink line, can only occur in the presence of oxygen, because oxygen has a strong affinity for electrons. And as those electrons have moved down and lost energy at each step, the low energy electrons are accepted by oxygen, which then will combine with protons in the matrix to form H2O. And so when we look at it this way, reminding ourselves that a byproduct of cell respiration is water, that H2O is formed by oxygen accepting electrons that have been transferred through the electron transport chain, combining with protons to produce H2O. So remember our overall equation of cell respiration. We start with our sugar plus oxygen, right? We'll overall produce carbon dioxide and H2O, and in the process, transfer the energy from the fuel molecules as the form of electrons, driving the proton gradient that will generate a huge amount of ATP. So we're trying to put the pieces together, break them down, and then put the pieces back together again. And this electron transfer will only occur in the presence of oxygen. Without oxygen, there is no final acceptor to accept the electrons, and they won't be dragged or pulled down this electron transport chain. So what is the actual movement? What do we mean by this electron transport chain? Each of those complexes are protein complexes. They're sitting and situated next to each other in the inner membrane, just like we saw in the video. The complexes themselves contain the actual electron carriers that are cofactors. They're metal ions, like iron ions, that can be reduced and oxidized by accepting or donating electrons. The sequential acceptance and donation of electrons will result in movement of hydrogen ions across the membrane. And each transfer of those electrons, they lose some energy. That energy is used to pump the protons across. So let's look at what do we mean by the actual movement of the electrons, and how does movement of electrons result in hydrogen ions being moved from one side of the inner mitochondrial membrane to the other side. So let's start with this example. We have an electron carrier that is in its oxidized form, so it will accept an electron. Here we have an electron that will be accepted by this carrier, and transiently it becomes negatively charged. It doesn't like being negatively charged, so it will accept a proton from water in order to balance that electron charge. And now our electron carrier, it's picked up an electron, but it's also picked up a proton, so now it has a hydrogen atom. Now it's in its reduced form. Notice that it's picking up hydrogen from one side of the membrane. Think of these molecules are sitting in a membrane. It's picking it up from this side, which happens to be the matrix side. And if we look at an electron carrier that is reduced such that it has a bond, it shares a bond with a hydrogen atom. When it's reduced, it can donate its electron to the next member of the electron transport chain, and it becomes, therefore, when it by transferring its electron, it becomes transiently positively charged. Well, it doesn't like being positively charged, and that positive charge, that extra proton, will be donated to water. But notice it's donated to water on the other side of the membrane. It's donated to the aqueous solution of the matrix. In that process of first losing its electron and then losing its proton, that electron carrier has become oxidized. So the protons are picked up on one side and they're released on the other side. And this is because of the way that the electron carriers are oriented in the membrane, that they become reduced by picking up protons from one side of the membrane, and they donate their protons to the other side of the membrane. So if we put these pieces together, here we have an example. Again, there's our gray bar is our membrane, our inner mitochondrial membrane. These colored shapes are different respiratory complexes sitting in the membrane, physically next to each other. And if we look at up here, we see that the green one is an electron carrier that's holding a high energy electron. If A transfers its electron to B, a proton is picked up from one side of the membrane, the matrix side. B then becomes reduced. When B transfers its electron to C, B becomes oxidized when B loses its electron, and then it releases its proton to the other side of the membrane, which is the intermembrane space. So when this type of electron transfer and picking up and releasing of protons to the other side occurs over and over and over again, we end up with a proton gradient across the inner membrane. Higher concentration of protons in the intermembrane space. So what we're showing here then is the coupling of electron transfer and creation of this proton gradient. This is where we'll leave right now. We will pick up on Friday, finish electron transport, correlate electron transport with proton gradient, correlate proton gradient with the synthesis of ATP, and then we'll summarize again all of the different, all of the yield from this overall aerobic respiration in the presence of oxygen. All right, so that's enough for today. I see a couple of questions in there. How does the electron lose energy again? I have office hours today if people want to ask me specific questions. Did you answer it? Okay, good. I do have office hours today from 1 till 2 if anyone wants to meet me there, or you can always ask questions by email as well. All right, see you on Friday.